All right. So, as I was introduced, my name is Malgorzata Gazda, and I was a graduate student at CBO University of Porto. And if you have any questions and interest, please contact me on Twitter or send me an email. And I'm very happy to show you my work focused on genetic basis of carotenoid ornamentation in bird parts of Canaries. So here in the first slide, you can see two portraits of red canaries. And the difference is that the first one has intensively colored red beak, and the other one has like sort of whitish beak. And then the one with the red beak is called Urucum canary. That will be the name I will be using for that presentation. And the story is that the breeders really wanted to create the red canary, which is on the right. So in general, the domestic canaries are yellow or white, but through the process of crossing with other birds, which is red siskin, they managed to create the red canary. And then spontaneously in the colonies of canaries, the birds with the intensive beak and also leg coloration arose, and that will be the story I will be showing you today. So this paper was published in Molecular Biology and Evolution, and here you have a QR code if you would like to access the paper. And I would like to say a couple of words why carotenoids are so important for birds. So first of all, there are, well, the carotenoid coloration, it's widespread in different species, and it's assumed to be an honest signal of the individual quality. But why is that? So the birds cannot produce the carotenoid themselves. They have to acquire them through the diet, so eating seeds, for example, and then processing it through the metabolism, and then eventually depositing them in the feathers or the skin, as here in the beak and legs as ornament. And that is also correlated with the stress and hormones. And actually having too much carotenoids can cause oxidative stress or also immune response. And Canaries is a really good model to look at the genetic basis of many traits. Here I focus on carotenoid coloration, but they can have different colors. So as you can see here, for example, red and yellow canary. They can have also weird shapes of the feathers, like for example, in a couple of breeds here. They can also have different postures, shapes of the body. And finally, they also can have different type of vocalization. So they were selected for different kind of songs. So this is like a great resource to look at many different traits in this animal model. And objectives of my study was to link the traits with high importance for avian evolution. So in this case, coloration and its genetic basis. So I asked myself a couple of questions. So are the genetic basis of the trait simple or complex? What are genes involved? What are the mutations involved? Are these coding mutations or regulatory variants? How did genetic variant arise? And finally, and excitingly, what are the biological implications? And to study that, I used different approaches. So first, I had to characterize the phenotype. So compare here you have the example of the yellow canary with white recessive canary, but in this study we will have the red canary with Urucum canary. And then, so for example, quantify how much carotenoids they have in different parts of the body. Then to do the whole genome sequencing and do the comparison between different breeds, looking at the population differentiation statistics, for example, here shown FST, to find the candidate region or regions if there was more than one involved. And then to follow up this kind of study, I usually try to do the functional validation. So for example, trying to express different isoforms of the gene in the cells and looking if, for example, carotenoids are acquired. And finally, try to understand the implications of the study. How does it relate with the plumage coloration or with the metabolism, the individual quality, and so on. And for this study, I used 2 data sets. So as I said, first one was the genetic part. So we did the whole genome sequencing of 2 canary pools, the urucum and red. We chose the red because that was the breed that the mutation arise. And then we followed that with the crosses and confirming that the region with linkage mapping. And finally, genotyping for IBD mapping. And the functional experiments that we did was, first of all, the HPLC analysis in feathers, beak, and retina. Then measuring the gene expression by qPCR and calculating the allelic imbalance. And finally followed with protein modeling and carotenoid assay. So first of all, we quantified the phenotype. So as you can see here, it's the wild type with this whitish beak. And then we quantified in different tissues. So in the graphs A, B, C, D, you can see first showed the wild type, and we have individuals from both backgrounds. So actually the urucum breed can be both yellow and red. Yes, so first it's wild type, and then you can see that in the beak there's hardly any carotenoids, whereas in urucum there are different levels of carotenoids, and then the ones with the yellow had, well, it was only 1 individual, but it had more carotenoids than wild type, but a little bit less than the red ones. And well, basically similar in the feathers. So in the wild type, there were less carotenoids in the yellow, and well, the red feathered birds, they accumulate much more carotenoids in the feathers. But you still can see that the urucum birds tend to have a little bit more carotenoids, although still the sample size is small because it's very challenging to sample the urucum birds. Actually, they are pretty expensive, and then they were not really fit, healthy, so that the breeding is a little bit challenging. So that's why we ended up with quite small sample size. And then interestingly, in retina, you can see different levels of carotenoid accumulation in the wild types, both in yellow and red birds, and like middle level in urucum, whereas apocarotenoids, so the products of disruption of carotenoids to smaller particles, you can see that there are quite some apocarotenoids in retina of wild-type birds, both yellow and red, and in urucum there's hardly anything. So I will come back to this finding later, but basically this means that the BCO2 is non-functional, and then the carotenoids are not disrupted, and this has implications for the vision. So coming back to genetics, I did the whole genome sequencing, and then I looked at the population differentiation across the genome, the FST you can see here, comparing the urucum birds with red birds, and you can see one clear peak marked here with the pink arrow. And in that region, BCO2 gene is located, so beta-carotenoid oxygenase 2. And that gene was already implicated to be important in carotenoid metabolism in other species. For example, related with the meat color, with fat color. So that seemed to us a good candidate for our phenotype. We were a little bit disappointed that it wasn't like different gene. That would be more exciting, but it seems that actually BCO2 is quite important gene in general for carotenoid metabolism. So I looked at the variants in that region, and I found one missense mutation. And here you can see the protein sequences of different species. And the one marked with the pink arrow, this mutation, it's only present in Canary, in Canary. There's no other species that would have such mutation. And if you look at the amino acids around, it seems that the region is pretty conserved. So there's not that much variance as in the other regions, a little bit farther, for example. And then to find out like how functionally important could be this mutation for the protein, we did the protein modeling with our collaborators, and we found out that so the change of the amino acid from arginine to histidine causes the structural distortion of the protein. And we hypothesized that this leads to the observed loss of the activity of the protein. And then to functionally validate this hypothesis, we did the assay, the carotenoid assay. So we had the cells that had either wild-type BCO2 or mutated BCO2 form. And here in the graph, you can see that, so the wild-type is in blue, and mutated BCO2 is in red. So in wild-type, there's not much substrate, so the cells were given the substrate, and then they should have processed the substrate to the product, and the product is the apocarotenoid, so the PIC1. And the PIC1 we find only in wild-type BCO2. We don't see that in mutated BCO2 or denaturated BCO2. So this means that the cells had the substrate of the mutated BCO2, but they were not able to disrupt the carotenoids, produce the product. So this led us to the conclusion that BCO2 lost its activity. So the implications from this study. Carotenoid cleaving activity leads to coloration gain, modification, or loss. BCO2 impacts conpigmentation and tune sensitivity of photoreceptors. And there is an interplay of the signaling benefits, physiological costs that may be mediated through regulation of BCO2. And I would like to thank to my supervisor, Miguel Carneiro, and other colleagues, collaborators, and everyone who contributed to my PhD BioDiff for the doctoral training, and FCT for the funding, and SMBE for inviting me to present this journal club. And I'm very happy to take your questions and discuss in more detail whatever you would like to learn about the study more. I noticed there were some things in the chat, but I didn't. Oh, okay. People introducing themselves. Okay.
Thank you very much for your presentation, Malgorzata. It was very clear and nice. If anyone has any questions, you can post it in the Q&A. I did already have a small question myself, more maybe an experimental one. You got really clear signals for your BCO2 gene, for example. And I think this relates to the two pools that you used. I was wondering how many samples from different canaries do you need in your pools to get?
Can you repeat?
Yes, so I was wondering how many canaries per pool you actually need to get a good sample size to get such clear signals for your genes that distinguish the both populations.
Okay, so, well, we can come back to the FST. So the thing is that, so the Canaries breeds are basically one species, but like, well, it depends which wild species you would compare, but sometimes you get much lower FST between the species than we see here in the Canaries. But so we know a little bit our model, and then sometimes it's a challenge because we get like too many peaks across the genome, which not necessarily are related to the trait we want to look. So it's very helpful that there is a lot of knowledge about like genetic bases coming from the canary breeders. So for some studies, we know that if it's gonna be the autosomal gene, or is it, or the trait it's sex link. It's also useful to know if it's recessive or dominant, and this sometimes we also know from previous work performed by the canary breeders. And then sometimes it's quite useful to it's not, in this case it was a good idea to have only urucum and red canary to have a nice clear peak, but sometimes it's quite good to play around with different breeds to see if, for example, so if we have the red birds, quite often we get, well, we always get the peak that is related with the red coloration, and that is why, because the red coloration was introgressed to Canaries, so the signal in the genome is very strong. Here we don't see that one because we have two breeds that are both red. So that's how we managed to remove that peak. And this is the way, a little bit like how to understand the differentiation across the genome in different breeds related to different traits. Although for some traits it might be quite challenging, and also like, well, one thing is the selection, but there's like also a lot of like random, you know, the genetic drift and demographic processes that also can create signals in the genome that are not related to any specific trait or anything. They just happen to be there because for some reason, well, randomly the genome, well, the part of the genome was differentiation. So differentiated. So sometimes it can be quite a challenge to try to find a clear peak in your data. The other thing to do with that is to try to sequence more individuals, so that then you have more power to try to show your differentiated region.
But you can't really say like this is the number of individuals you typically should go to. It really depends a lot on the situation, I guess.
Well, the number of individuals really depend on trait, and then so this is, well, so I have another project on the trait that is that is dominant. So the recessive traits are easier, but the dominant one, the individuals can be heterozygotes as well, and that makes it much more challenging to find out the region, and the signal is much weaker. So then this means that it's needed to sequence more individuals, but it's really, it really depends.
Okay, thank you. I have another question here from Christoph Demsimos. He asks, what part of your nice work could be automated in a tool?
Well, that would be great. So, well, probably part of it could be created a pipeline. Well, but it really depends. So for this project, for example, I used pool data, but for it depends on the project. Sometimes we use the individual whole genome sequencing, and then the tools and the approaches are really different than the pool data. So that's one of the challenge. And so the genomic part, I think it could be, well, at least parts of it could be like a little bit more automated, but sometimes it also requires like checking the quality and trying to find out the balance of like being not enough strict, so like having too many low quality SNPs, or being too strict and then having basically no data. So it might be dependent on, but it also depends like how many individuals were sequenced and what was the coverage. And then for the phenotypic characterization, I don't think there's any way to make it automatic, because it really depends on traits. So if it's carotenoids, then it's maybe not too challenging, because you do HPLC and measure the carotenoid content. But if you want to look at shape of the body or at feathers or singing, then you need to design completely different assay, how you try to characterize the phenotype. And then the same applies to functional validation. So it really depends like what part of physiology is it related, and like what will be the idea how you try to validate your funding. So actually the phenotypic characterization and then the functional validation are much more challenging than, you know, doing the genome scan. The trick with the genome scan is like to come up with a good model. So you compare, well, to try to find two breeds that they differ in the trait of your interest, but not too many other traits, because if they differ, if one has melanin, other has carotenoids, one is yellow and one is red, then there will be a lot of peaks across the genome, and that will be not ideal study design. So the short answer, it really depends.
Okay, thank you. There is another question from Aida. Are there differences in the coloration that are related to the sex of canaries?
Yes. So we published recently another story, and that is looking at the sexual diffromatism. So basically there is another breed that is called mosaic canary. And the mosaic canaries are characterized by the fact that the males have a lot of carotenoids in, for example, in the head and in the tail, and the females have much less carotenoids. And then actually, interestingly, we showed that the same gene, so BCO2, is regulating locally in the well, tissue-specific manner the amount of the carotenoid that is accumulated or disrupted by the bird, and this is somehow related. So it's probably somehow related with the hormones, but we haven't really look at it ourselves, but it will be very cool to look like how the hormones regulate BCO2 expression and how it works on the like more cell mechanism way. And this study was recently published in Science.
Okay, she asks, are there any, so she says, thank you. Are there other candidate genes that could be involved?
In this red big phenotype? I don't think
Yes.
so. I think it's BCO2 that is either functional or not, and then but the other, so now we know it's a missense in BCO2, missense mutation in BCO2, and then the gene is non-functional. But it would be very interesting to try. So we did a little bit. We look at the retinas and we look at the tissue, but I think so the breeders reported that, for example, Urucum Canaries, they had like problems with health, vision. No wonder that, well, they don't process the carotenoids. And then also reproduction. So I think that would be an interesting like follow-up study to try to find out what is like what this gene does more than just disrupting the carotenoids, because I think it is quite important for animal fitness.
Okay, she thanks you for the answer.
Thank you for questions.
I think there are no more questions here. Let's wait a few more seconds if any more questions come in. So I think no more questions are coming. I would like to thank you really a lot for this very nice presentation and all your replies to the questions, Malgorzata. And I would like also to thank all the people that asked questions, because really this promotes the discussion. Thank you, everyone, and hope to see you for the next webinar.
Thank you for having me here. I really enjoyed presenting. And if someone comes up with some ideas, questions, or anything that you would like to follow up, please reach me out on Twitter or by email.